Hi, welcome. I'm going to introduce myself today. My name is Dr. Joel Wertheim. I'm an assistant professor of medicine here at UC San Diego. I've been here at the AVRC for the last 10 years this month, but for those of you who don't know or recognize me, it's because my group hides off in a little corner that when they give tours, they rush everybody past very quickly to the more interesting parts of the building. Um, that being said, we have our own little fiefdom there, and today I'm going to share some of the work we've done on HIV molecular epidemiology and its relevance to public health. All of this work is done in either collaboration with the Los Angeles County Department of Health or the Centers for Disease Control and Prevention, with whom I serve a consulting role. So to give you just the most basic introduction to what molecular epidemiology is and what it can do and how it can be extremely powerful in understanding how HIV transmits in a population, I present to you a study we published in 2017 looking at the genetic distance separating the virus found in 756 named partners, named partner pairs. So we have the virus from an index case and their sexual and injection drug using partner, and we ask how many nucleotide substitutions separate the virus in the index case and their named sexual partner. And we can see that it forms this lovely bimodal distribution where we have this blue mode representing pairs that have highly similar virus, so similar, in fact, as to imply a direct or indirect epidemiological connection. And then we have another set of pairs shown here in the red mode, where we see virus that is essentially random with respect to genetic diversity. If you pick two people in New York City who are both living with HIV, sequenced their virus and said, how divergent are these viruses? They would fall somewhere in this red mode. Virus that diverged sometime in the 1970s and has been evolving away from each other ever since. Now, this means that even among named sexual partners, we can differentiate people who likely have a direct or indirect epidemiological connection and those that don't. And it's that genetic distance which essentially is going to found the entire basis for what I'm going to present to you today. So I hope that made sense. By identifying these partners with virus that is very, very genetically similar, implying a direct or indirect epidemiological connection, we can go into, say, a county-wide HIV surveillance database and identify clusters of individuals whose viruses all imply a direct or indirect epidemiological connection. And those are shown here for Los Angeles County, where last year a former postdoc of mine published an investigation into transmission risk and how it affects being a member of one of these clusters in Los Angeles County. She found that certain groups were more highly predictive of clustering in this network, indicating a higher risk of either transmitting or acquiring HIV. We're not talking about directionality here. We're just saying there's a higher risk that that individual was involved in a transmission event. Again, we're not distinguishing here between source and recipient partners. And what we found is that the group that had the highest odds of clustering in our network were transgender women, both those who injected drugs and those who had a sexual risk factor. And we see that they were among the highest odds of clustering, indicating that Los Angeles County should increase focus on this population, which up until this point wasn't even well characterized in their surveillance database. We also found that people tend to cluster with others sharing their reported transmission risk, but that different risk groups clustered more, as we would say, assortatively in the network. That is, people with one transmission risk clustered with people with the same transmission risk. So MSM tended to cluster with other men who have sex with men. Cisgender individuals who reported a sexual risk factor tended to cluster with each other more than they clustered with others. And transgender women who reported a sexual risk tended to cluster with each other, although not to the same extent as cisgender individuals or MSM. Notably, people who inject drugs did not cluster assortatively in the network, implying that at least in Los Angeles County, people who inject drugs, although they may associate with other people who inject drugs, aren't transmitting preferentially to other people who inject drugs, which, as we've also seen in other major metropolitan areas in the United States, like New York City, implies that harm reduction programs are effective in preventing transmission through needles. So how does this all translate to a public health context? What can we actually do with these data? So I'm just going to front load this slide here. Well, let's say we have somebody who is newly diagnosed in San Diego County, Los Angeles County, New York City, really anywhere in the United States. Well, we can do traditional name partner elicitation protocols where you can identify HIV negative partners. Of course, those people can be put on linked to PrEP. If they're found to be newly HIV positive, then they're diagnosed and we can help them progress through the continuum of care. But using the genetic network, we can also identify this group over here who are not named partners but are genetically linked. So the virus is telling us that there's an implied direct or indirect epidemiological connection. But they don't have to name those partners. And we can identify people who are, say, genetic partners who are out of care. Well, they can be prioritized for return to care services. Those who are viremic can be prioritized for suppression support services or other activities that could help them maintain viral suppression, whether that's transportation support, housing support, etc. And then if we find genetic partners who are in care and suppressed, well, there might be nothing that the public health department can offer them, so they don't need to be prioritized. But this is sort of the general overview of how a public health response could work when we have someone who's newly diagnosed with a genetic sequence on a cluster. So now I'm going to present to you some work by a postdoc in my lab, Britt Scathen, who investigated the geography of these transmission clusters in Los Angeles County and how they relate to the genetic clusters. So essentially, we're going to ask how HIV genetic clusters relate to time-space clusters of HIV diagnoses. So Los Angeles County, for those of you who dare to venture up there, is very large, actually, like most counties in California, and it can be divided a whole host of different ways. They have their service planning areas, where all of Metro LA is contained within one service planning area, and we have the South Bay, Southeast, and the like, but we also have these health districts where we further break down Los Angeles into what are not quite neighborhoods, but certainly more focused, so we can break it into 26 unique partitions, or we can go and we can break LA down into its 507 constituent zip codes. So we can get quite granular with the data we have, looking at where people were residing at the time of HIV diagnosis. And if we just look at these clusters in Los Angeles for people who resided in Los Angeles at the time of diagnosis and reside there today, and look at what SPA they live in. SPA is pronounced spa in Los Angeles. Guess that makes sense. And we can see that there's some geographic pattern in these clusters. We see this one here that has a lot of red in it, which is Metro LA, where most of the cases are anyway. Here we see quite a few cases in Antelope Valley and San Gabriel, so a lot of yellow and a lot of pink. But we don't see any cluster that is just one SPA. Although we definitely see some geographic signal, it's not that here's a metro cluster, here's a South Bay cluster, here's a West cluster. So what if we look at these at individual level? So if we take 3,701 people who were clustered in the genetic network who resided in Los Angeles County at the time of their diagnosis and since 2010, 64% of them were diagnosed, had a genetically linked partner diagnosed in the same year. So those are people who were genetically linked in time. 19% had a genetic partner who were diagnosed in the same zip code. So those are people who are genetically linked in space. Now, if we ask what proportion were genetically linked in time and space, it's only 7%. So only 7% of all clustered individuals in Los Angeles County were diagnosed in the same zip code and the same year as their genetic partner. Now, we could break this down into quarter, we could go up to health district, or we could go to SPA, but time and time again, we just don't see a very, very strong signal for geographic and temporal overlap in these genetic clusters. That being said, this 7% is over 5 times more than you'd expect based on the null distribution of when people were diagnosed and where they were diagnosed. But it's still a very, very small number, indicating that when we see clusters in time and space, they're really not indicating underlying transmission events. And if we look back, Susan.
You didn't actually go the other way. In other words, you went genetic clusters are not necessarily time-space clusters, but did you look at time-space clusters to know that they're not? What proportion of those are genetic?
Did we
The
pull that
inverse.
out? No.
Yeah.
Britt, make a note, pull that out. It's not going to be, at least in LA, I don't expect to see. Yeah, no, that's a good point. Okay, so what if we look just assortativity in the network just by geography? Well, the most assortative factor in the network is race ethnicity, which is probably not that surprising. The next most assortative factor that we looked at is actually service planning area. So the network is fairly assortative by these large geographic areas. Then transmission risk, then health district, then age of diagnosis, and then what is barely assortative but significant because we have thousands of people is zip code. So the network is assortative by geography, just not extremely meaningfully. Not enough that if you saw 2 people who were diagnosed in the same zip code in the same year or the same quarter, you would necessarily want to make much of it. So then we decided to do, Britt decided to do a little fancy analysis, which is to take everyone in genetic cluster who clusters genetically, and then everyone who clusters by their health district. So everyone in one health district goes in a bin over here, and then everyone in a genetic cluster goes in a bin over here, and we look to see in this nice happy Venn diagram what the overlap between those two are. And using this, we can get sort of this individual metric of what association exists between genetic clustering and health district. And we can go start to look at our other risk factors, whether it's race, ethnicity, age, or transmission risk. And she computes this nice little statistic called the Jacquard index, which is just, for those of you who are math phobic, taking this little slice over here, which is the intersection between these 2 circles, over the union of the 2 circles. Intersection, union, everybody remembers 8th grade math. I don't. But you can put that into a nice general linear model, and you can look at which risk factors are higher and lower in this Jacquard index, who has more overlap between their genetic and geographic cluster. And younger people, maybe no surprise, tend to have more geographic overlap with their genetic clusters, decreases as you get older. This could also be a function of age diagnosis, of about how long you were infected at diagnosis. Although we did correct for CD4 in this model, there's still that trend of younger people having more local partners. We find that cis men who did not report injection drug use or sex with other men were more likely to be assortative in this network geographically, but trans women actually had less overlap between geography and their genetic cluster. So they were essentially geographically over-dispersed in their clusters. And we found that African Americans were also geographically over-dispersed in their clusters, which may reflect the geography of Los Angeles itself. It means that, so for example, African Americans and trans women, members of their clusters are less likely to be found in their same health district. So there's less intersection here for those groups than you would expect, at least compared with, say, MSM, or for African Americans compared with Latino or Hispanic individuals. Yeah, cis men, this area is overrepresented. compared with MSM, who I believe are the reference group here. Okay, so that is our overview of geography. Now we move on to interesting analysis by Adiba Hassan on the relationship between these HIV transmission clusters and the care continuum, which I haven't seen anybody look at before. So I think this is kind of interesting. So we want to know, how does growth of these genetic clusters, so we have a rapidly growing clusters and we have slow growing clusters, how does that predict progression of individuals through the HIV care continuum? So just to go back to some work that we published earlier in New York City, we found that rapidly growing clusters, which are identified here in red, every point on this line is 500 individuals in a rapidly growing cluster in New York City, and the y-axis indicates the number of future genetically linked cases that are linked to that individual in the following year. So these are people in the most rapidly growing clusters in New York City, and how do they predict new HIV cases coming into the surveillance system? And if we compare that to 500 random cases in the surveillance system or 500 random clustered cases in the surveillance system, how do the most rapidly growing clusters compare? Well, they predict more future growth than random nodes or random clusters, which tells us that rapidly growing clusters give rise to more HIV cases than non-rapidly growing clusters. So growth in this network past growth in this network predicts future growth. So what is notable about the individuals in these rapidly growing clusters, and how do they engage with the healthcare system? So we divided the Los Angeles County network into 3 groups: people who were in slow growing clusters at diagnosis, essentially, I believe, unclustered. This is then we have people who were in clusters, but those clusters were very slowly growing. And then we have people in high growth clusters, people who were diagnosed in the most rapidly growing clusters. And we asked, using a Cox proportional hazard model, how long it took them post-diagnosis to be linked to care, how long it took them after they were linked to care to become virally suppressed, and how long it took them after they achieved viral suppression until they experienced their first viral rebound above 1,500 copies per ml. And we use this based on lab reported data. So every viral load in CD4 count that was run and reported to Los Angeles County Department of Health served as our outcome in this study. So first, people who were in the most rapidly growing clusters. So sorry, I'll take a step back. We might think that people in the most rapidly growing clusters progress the slowest through the continuum because they are most likely to give rise to future cases, and not being diagnosed, not being linked to care, not being virally suppressed, or not maintaining viral suppression all could lead to onward transmission of HIV. That is not what we found. What we found is that people who were in the most rapidly growing clusters were actually linked to care slightly faster than those people who were unclustered or in medium growth clusters. There was no difference in the time from linkage to care to viral suppression. However, there was a slight increase in time from diagnosis to suppression for individuals in the most rapidly growing clusters. And our most robust result is that people who were in the most rapidly growing clusters actually took longer to experience virologic failure. So failure of viral suppression, which means people in the most rapidly growing clusters, in any way that we can slice up the data, either progress the same through the continuum or actually progress slightly better in that they're more adherent. So this could mean that rapidly growing clusters are just indicating people who engage better with the healthcare system. And that's partly why we see clusters grow rapidly. Susan looks very concerned.
Sorry, I really was paying attention. This is New York, right?
No, this is LA.
This is LA.
Yeah.
I was paying really close attention.
It's good to have LA.
So are these individuals prioritized? I'm not using the word target. Prioritized by the health department to a greater extent?
Historically, this is all 2010 through 2014, so they knew nothing of the network at this point.
Okay.
They might be prioritized for other reasons. Here, these models are just taking into account, say, stage of infection at diagnosis,
Yeah.
race, ethnicity, birth, sex, and gender, and transmission risk.
I mean,
But
that's my only question, is that individuals in the most rapidly growing clusters may have been actually prioritized by the health department for reasons independent of their cluster growth. In other words, more acute infection, et cetera.
yeah, so some of that is accounted for here. In the more rapidly growing clusters, yes, there would be. But they also, so for example, that slower time to rebound could reflect people who got HIV testing. If they were diagnosed early, they could be in a rapidly growing cluster, and then that means that they are just engaging with the healthcare system more. So yes.
So Joel, sorry, this is very ignorant questions here, but so in LA County, the county are keeping track by name of viral loads of everyone that's tested?
Yes. So just to take a step back on how HIV surveillance works in the US. Yeah. So every CD4 measurement and every viral load measurement is reported, and genotype is reported to a health department in the US. So regardless of what county or state or protectorate you live in, viral loads are mandated by law for name-based reporting. So yes.
They're
and that's been
mandated by
going-
law for name-based.
Yeah, so every viral load gets reported to the health department, so they can keep track to know who's out of care, and they can prioritize return to care services.
Is this the same in San Diego? You said it's.
Yes. Yeah. I could give a whole 4-hour seminar on what I've learned about HIV surveillance in the US in the last 10 years.
So could it be something in the biology of the virus that this really big cluster, it's a virus that's better to reproduce, better to pass from one people to another, one person to another, and better to mutate against viral antivirals?
I plant questions with my postdocs. That's part 3. So viral biology is coming up next. Whether or not sort of virologic sort of suppression failures related to that, or time to suppression, of course, people with higher viral load could take, just based on viral kinetics, is going to take slightly longer to become virally suppressed. I don't think that's what's at play here. The reason is that most of this actually, so the time to say viral suppression is really based, that entire difference is based on whether they got linked to care within 2 months or not. So people who got linked to care within the first 2 months, no difference in time to viral suppression based on when they were diagnosed to first viral load under 200. People who weren't linked to care within the first 2 months after diagnosis, those people in high and medium growth clusters became suppressed faster. So I think what we're looking at here is really about behavior and engagement with the healthcare system. It could also be the healthcare system or the public health system engaging with those individuals. But given that this molecular work was not being done in LA in 2010, we're sort of looking at the totality of the behavior. There is one more interesting thing that we found here, and potentially more interesting than whether or not people in rapidly growing clusters took longer to get to viral suppression, and that comes from a gamma frailty model, which, if you want to know what that is, Adiba's in back and she can explain it to you. That's why we keep her on. Basically, it is a way of incorporating random effects into a temporal model. And essentially, cluster membership, the simplest I can say this, is a random effect governing how quickly you progress through the continuum of care. So that means that whether or not it's moving from time from diagnosis to care, time from care to suppression, diagnosis to suppression, or suppression to rebound, you are going to move to each event more similarly to your genetically linked partners within a cluster than across clusters. Now that's after correcting for things like risk factor, race, ethnicity, age of diagnosis, stage of diagnosis, geography. You're going to move more similarly to your genetic partners than to everyone else. Now, your genetic partners are also your sexual and social partners, or at least partners of partners, which means if there's a cluster that is progressing historically very, very slowly through the continuum, people who are failing to achieve viral suppression, or once they get viral suppression, there's a failure to maintain suppression, their genetic partners, we can predict, are more likely to do that. Now, we don't know about the timing and how actionable this is, but we do know that within a cluster, there is signal based on your genetic partners of how long it's going to take you to progress through the continuum. So there's meaning there. Whether or not it's actionable, we're still working to find out. Yes.
Do you think the longer the suppression is a factor for the virus?
I mean, higher viral load could have an effect. We're looking at we're only sensitive to months here, so we have
Right.
month of year. So I mean, I think it could matter for weeks, but I don't know if that's much what we're seeing, and whether or not there's certain viruses that aren't being suppressed. I don't remember if we looked at drug resistance in these data. No, we did not. That's interesting.
That was going to be my question, because if there is not significant resistance, even in the acutes, our mean time to suppression is 30 days. So I don't think viral load is the explanation.
Okay. Yeah, and like I said, mostly what dictates your time to suppression is your time to your first CD4 viral load post-diagnosis.
Did you have any socioeconomic status data or health insurance data? I mean, I guess zip code could be a surrogate for
so
socioeconomic status to a certain extent.
We did incorporate socioeconomic data based on a question Susan asked us about 6 months ago. So we took percent living below the 100% federal poverty line within a zip code or health district, the zip code of diagnosis, and no effect there. I mean, I should say the frailty was robust to that inclusion. So it's not there's. I guess what we're saying is that there's more here than just. But of course, even within a zip code, in really any place, you can have high and low SEC, and but in our attempts to correct for it, we still found this signal. I guess that's the best way to put it. Okay. So people in rapidly growing clusters do progress faster through the continuum and maintain viral suppression longer. People in the same cluster progress at similar rates through the continuum, and we might be able to use molecular epidemiology to prioritize clusters for people more likely to experience viral rebound. They may be people to keep track of if we see that rebound is very common in those clusters as one possible translational outcome of this study. All right, now we're going to get a little different, but I think this is work that I'm very proud of and I wanted to share with you today. And it's looking at the viral characteristics, looking to see if we can detect natural selection favoring more transmissible HIV in the US molecular transmission network. We are just getting ready to submit our final round of revisions to Nature Communications, so hopefully this will be out in press soon. Okay, I've told you that molecular epidemiology can identify high-risk subpopulations. For the most cynical among you, those high-risk subpopulations that we published on in New York City were young, recently infected, white men who have sex with men. That is not exactly the most insightful finding I could present to you, that in New York City, these are high-risk groups. So what else is going on? Is there any sort of viral genetic component to the variation in the transmission rate across this network? And we think that the answer is yes. So the frequency of clustering in a molecular transmission network can be indicative of relative transmission rate. Now, My training, despite the fact that I've been living in the Department of Medicine for the last 10 years, is actually as an evolutionary biologist. So when I see this, I think of this as fitness. This is whether whether a strain gives rise to more strains in the future than another virus, and that is differential reproductive success. And in classic evolutionary Darwinian terms, that is fitness. Now, the first time we decided to look at viral fitness, we went to where the pickings were pretty easy, and we looked at drug-resistant mutations. Now, drug-resistance mutations can affect viral replication within an individual, but they can also affect how well virus transmits between individuals, and that between individual transmission and the frequency of that essentially is the viral population fitness, which determines the frequency of a mutation in the population. So we went and we looked at M184V, a very rare, relatively rare drug resistance mutation that is strongly deleterious for replication in vitro within an individual in the absence of drugs. And if we look at the clustering frequency, how often did two people with V at position 184 cluster with each other in the network? Well, 6 times. How often did somebody have a V but was clustered with somebody with an M? 163 times. So their relative clustering frequency in the network was only 3.6%. Compare that to people with M, where their clustering frequency with people with wild type virus was 38.9%. That means that having V at 184, you have a relative transmission fitness of 0.9, which is a huge selective disadvantage, which is why we don't see clusters of M184V in the US. We see individuals that occasionally will transmit that, but I can tell you in all of New York City, there is not one legitimate cluster of transmitted drug resistance of M184V. It just doesn't progress through clusters. We can see a handful of pairs in ART naive individuals, but that's it. What about K103N, an NNRTI resistance mutation that I think for a large part motivated the switch to NRTIs and integrase inhibitors? And a good reason for that is because not only does it persist within an individual, but it actually persists in the population. So people with N at position 103 cluster at a frequency of 37.1%. People with K cluster at 38.5%, which gives K103N a relative transmission frequency or fitness disadvantage of only 0.97, indistinguishable from 1, which means its fitness is essentially no different from wild type, which is why if we look at these clusters of K103N, and you can see people with K103N are colored in blue, people with K103S, a related mutation of similar effect, are colored in purple. Anyone who's marked with an N is ART naive. Only people marked with E are ART experienced at diagnosis. You can see that this cluster represents transmission of K103N going on for a decade in the United States, uninterrupted among ART naive people, which means once this mutation arose here, it just continued to transmit to people regardless of whether or not they had drug at diagnosis, which is why we moved away from this mutation, because its fitness is essentially, once it gets in the population, it's very difficult for it to go away. That could be due to a variety of compensatory mutations, but the point is this mutation persists in the population. Now, if we go back to, say, a protease inhibitor, L90M, the situation was actually even worse. 48.4% of people with an M clustered with someone else with an M. That's higher than wild type, which means strains containing L90M were actually more fit than wild type virus in the US population, which means we have clusters persisting to this day of L90M that originated with the introduction of the drug. So here this goes all the way, we have a cluster of 80 people going back all the way to 2001. Almost none of these people had been exposed to ART at diagnosis, but the cluster persisted, and that's why we don't use protease inhibitors. I think this also, in the paper, we make the argument that this is an argument for moving away from PIs and NNRTIs in places where drug resistance testing is less frequent, like sub-Saharan Africa, and we should just switch over to NRTIs and integrase inhibitors as first line everywhere. because, well, the evolutionary biology is on our side. Okay, now we did all of this just using that genetic distance-based clustering, which, as enthusiastic as I am about it, is a very, it's quick, it's dirty, and it is not at all sophisticated. Not like the Swiss cohort, who went in and after we published our results, went and did the same study in Switzerland using a very complicated phylogenetic birth-death model approach, which I won't get into today, other than to show you that they got the exact same results that we did using a far more sophisticated model. Here, M184V, lower fitness. Here, K103N, indistinguishable fitness. Here, L90M, higher fitness. Our paper came out first. This paper was published in a better journal. Or maybe virus evolution is the journal of the future. Who knows? Anyway, but I only say this to tell you that relative clustering in a transmission network can be used to estimate fitness, even though we're using a very crude genetic distance-based approach. It agrees with the far more sophisticated phylogenetic approach, which really isn't applicable to data sets of, say, tens or hundreds of thousands of individuals. Which brings me to the viral component I want to talk about, which is set point viral load. So set point viral load is after initial infection where you get a spike in viremia. It is where the virus plateaus during stage 1 infection. Now, for some individuals, it can be high. For some individuals, it can be low. And then eventually, after stage 1, you move into stage 2, and then stage 3, AIDS and viral load increases. But for a very long period of time, this set point viral load is relatively stable in the within an individual. Now, set point viral load also varies dramatically across individuals. So some people have a very low set point viral load, say 10 to the order of 10 to the minus 3. Others can have orders of magnitude higher, 10 to the 5 copies virus per milliliter of blood. So we're talking about anywhere between 1,000 and 100 or 500,000 copies within an individual. And this is a relatively stable metric in the absence of antiretroviral treatment. So we have extreme variation in a population. Viral load in the absence of ART is also highly predictive of disease outcome. So your duration of time that you're AIDS-free decreases the higher your viral load. So with people who are very low viral load, it can be up to an average of 20 years between infection and AIDS. Whereas for people with very, very high viral load, it can be much, much shorter, on the order of just years. But also infectiousness, or the probability of transmitting per event, is much, much lower in people with low viral load and higher with higher viral load. Essentially, the more virus, the less likely you are to transmit per event, but the longer you are infected, so the more opportunities you will have, or the more time you will have to prevent transmit before AIDS. Which leads to this expected curve, which says for people with low viral load, you have long survival but low infectiousness. For people with high viral load, you have high infectiousness but short survival. And that there is probably some optimal intermediate viral load that optimizes the number of opportunities for transmission and the probability of transmission per event. And that from an evolutionary perspective, the virus may have attained this optimal transmission fitness or intermediate viral load, and we just see the residual variation in the population. So infectiousness, just to distinguish, is the probability of transmission per sexual event, and transmissibility is the relative number of onward transmission events over the course of the entire duration of viral infection. Okay. As I mentioned, I'm an evolutionary biologist, so I think about this in evolutionary terms. Now, natural selection, or the change in a population, must occur if a population is variable, if that variation is heritable, which means there's an underlying genetic component to that variation in the population, and whether or not that variation leads to differential reproductive success or a different frequency of transmission of this virus in the population. Now, I'm not going to touch the heritability question. I think it's been answered, so we're just going to skip over that. And we've already established that there's variation. So I'm just going to focus on whether or not viruses with higher or lower viral load have differential reproductive success over the lifetime of the infection. Now, there are plenty of papers talking about what might happen. So there was a paper published out of Seattle University of Washington group that said under antiretroviral treatment, test and treat, where everyone is goes on therapy immediately after diagnosis, you would expect selection for an increase in mean set point viral load, because if you take away time to AIDS, all that's left is infectiousness before diagnosis. So that would create selection to increase set point viral load. In contrast, a group in the UK has estimated a potential decrease in transmissibility and virulence in Uganda, where there appears to be a replacement of subtype D with subtype A. So whether or not this is a differentiation between the subtypes or the opposite evolutionary trend, it's not really clear, but we do see evidence for a decrease in transmissibility and virulence in Uganda. What about in the US? So we queried the US National HIV Surveillance System, which has genetic sequences from over 250,000 unique individuals as of 2016. And we found 41,000 individuals who were ART naive at diagnosis, genotyped within 3 months of diagnosis, had a viral load within 4 months of diagnosis, were subtype B, and had no evidence of perinatal exposure. 24,000 of these individuals had no evidence of drug resistance mutations or wild type. So we went from about 250,000 people to 24,000 pretty quickly. And then we asked, do they cluster in the network? Are they genetically linked with another wild type virus? Because we wanted to remove the effect of drug resistance from this entirely. So we went and we built our network. And we looked and we divided people into stage diagnosis. We looked at people who were stage 0, those with a negative HIV test in the last 6 months. Stage 1, CD4 count above 500 at diagnosis. Stage 2, CD4 count above 200. And stage 3, CD4 count below 200 or evidence of opportunistic infection at diagnosis. And really what I want to focus on here is those 6,000 individuals who were diagnosed at stage 1 with their set point viral load, a median viral load of about 19,000 copies per milliliter, which clustered at a frequency of about 47%. Now, of course, you'll see here that as initially people have higher viral load diagnosis and it's low, and then it increases depending on staging, which is exactly what you'd expect. And the frequency of clustering goes down as the time since diagnosis goes up, and that would be expected because genetic distance increases over time, and people here were infected for a maximum of 6 months at stage 0, where in stage 3 they could have been infected for over a decade before being diagnosed. Okay, people diagnosed during stage 1 infection, if they were unclustered, unclustered individuals had a median viral load of 19,000 copies per ml. Clustered individuals had a median viral load of 22,000 copies per ml, which means if we compare them in a multivariate regression model, we see that there was a slight 1/10 of a log, but highly significant increase, benefit of having thousands of people in your data set, increase in viral load for people who were clustered at diagnosis. Okay, so that's one measurement in one particular population. How robust is this effect on viral load on clustering or transmissibility? Because that's highly significant, but potentially not robust. Okay, what if we look across all the stages of infection and diagnosis? Well, for people at stage 0, there's no difference. Likely because we only had 400 people there, and also because viral load during acute and early infection, as Susan will tell you, is highly dynamic. It can change over orders of magnitude in the course of weeks. But for people at stage 1, clustered individuals have higher viral load. At stage 2, people who are clustered have significantly higher viral load, again, 1/10 of a log higher. And for people with stage 3 infection and diagnosis, we also see a significant increase in those who are clustered versus not. What about when we start to adjust for all of the risk factors I've been talking about, whether it's birth, sex, transmission risk, race, ethnicity, age of diagnosis, CD4 count, diagnosis year, we see that this effect is robust to the inclusion of the standard surveillance covariates, including in both stage of infection and CD4 count within that stage of infection. We still see a robust association. Even after accounting for diagnosis year, because clustering has gone up over time, but actually people are getting diagnosed earlier and earlier over time, which, except for that small proportion of people diagnosed with acute or early HIV, actually results in lower viral load. Okay, so if we build our network at 1.5% genetic distance cutoff, which is fairly conservative, we see this effect. If we build our network at ever increasingly conservative genetic distances, we see that this effect is robust. So these are people who we can be more and more confident are direct or indirect epidemiological partners, and transmission happened more and more recently. People who are diagnosed, people who are clustered have higher viral load than people who are not. Now, I think one of the, so all of this could be confounded with the factors that lead to clustering. But what we see is that people who are more connected in the network, so for every degree you have, for every genetic partner you have, your viral load, if you're diagnosed at stage 1, goes up. Now this is going from 0 to 1 to 2 to 3, 4, all the way. Every connection indicates a higher viral load. This is true even if we exclude unclustered individuals from this analysis. So we only look at the clustered people and say, do you have 1 degree, 2 degree, 3 degree? That delta is highly significant in all these models. And if we look for people as they have more connections, their viral load at diagnosis goes up. The same is true for cluster size. So people in clusters of size 5 or more versus clusters of size less than 5, clusters of size 10 or more versus smaller clusters. Viral load is higher in these larger clusters. Now, larger clusters tend to be older, and yet still we see, because it takes time for people to accumulate in those clusters, we still see higher viral load in those in large clusters versus not, which would be contrary to expectation, because as people get diagnosed earlier and earlier in infection, they will have lower and lower viral load over time. Now, this association, again, is robust to whether or not we include or exclude people who inject drugs in the analysis, because people who inject drugs, viral load wouldn't be expected to influence transmission through needles, but given that in a lot of settings for a lot of these individuals, even people who inject drugs are transmitting through sex, we don't detect any effect there. Also, if we only look at diagnoses post-2011, this finding is still robust. It's even robust to network structure when we just throw out all of our statistical models and just do a permutation test. We can see that this would be the expected difference in viral load between clustered and unclustered individuals, and here is our observed difference. So we see a significantly different observed versus expected for individuals diagnosed at stage 1, stage 2, and stage 3, but again, not in stage 0 because numbers are smaller and viral load is more dynamic. Okay. So natural selection must occur if a trait has variation, viral load has variation, heritability, a lot of other studies have demonstrated that it's heritable, and differential reproductive success. Well, we find that clustering, an indicator of differential reproductive success, is individuals with higher viral load are more likely to cluster and vice versa. So do we see this effect over time? Well, if we plot out all the viral loads at diagnosis for people diagnosed with stage 1 infections since 2005, we see yes, viral load has increased in the US at diagnosis by about 0.2 log since 2005. So in 2007, median viral load was 13,000 copies per ml. I picked 2007 because that's when the data really started to come into the national surveillance database. In 2006, it was up to 22,000 copies per ml. So we do see evidence for a trend in increased viral load over time. And we see differential reproductive success for individuals with higher viral load. This trend is robust for, we see the same pattern, but just a higher a higher viral load for individuals with stage 2 or stage 3 infection, but the association with time is still robust. Okay, so there are a lot of potential biases that could be influencing these results, and I'm going to take the last 5 minutes to tell you why I think we're robust to them. So first, recency of infection at diagnosis is associated with both higher sampling frequency and inferred transmission rate. But outside of stage 0, this effect would bias towards the null, in that people would be diagnosed with lower viral load, but be more likely a cluster. And we see the opposite in our results. Also, you would not expect this recency of infection to be progressive across the network as clusters get bigger and people become more connected. Shorter genetic distances between sequences is associated with less time since transmission. It's true, but again, outside of stage 0, this effect would bias us towards the null. More recent transmission, lower viral load. Again, this would not be progressive with continuity across the network. Year of diagnosis is associated with a trend in higher viral load. That's true, but the results are unchanged when considering year of diagnosis. So our regression model is robust to this covariate. Higher viral load can cause more rapid progression to stage 2 infection. That is true, but if someone were more likely to cluster, this effect would again bias us towards a null because we would put them in stage 2, but they would have the viral load of essentially somebody in stage 1. Even though it would be slightly elevated, it would be less than an average in stage 2. Importantly, we also, as reviewers pointed out, we have a lack of data on STD co-infections, but despite the fact that HIV tends to spread in populations exposed to already infected with STDs and vice versa, so they're predictive of each other, they don't transmit across the same time and space as HIV. So if you look at a cluster of people with hepatitis C and you look at their HIV sequences, that transmission pattern isn't the same. And if you look at people with HIV and look at the hepatitis C pattern of transmission across them, those transmission patterns are the same. So although we see an association between STDs and HIV, they transmit over different times and spaces. So you wouldn't expect that effect to be progressive across network connectivity. So what we report is that HIV subtype B in the United States appears to be under natural selection to become more transmissible and, in the absence of ART, more virulent. The strength of this selection, although is quite weak, for example, an entire log change in viral load would increase the odds of clustering by only 1.12 in our model, which means we're probably seeing about like a 1.2% fitness advantage in viral load. Also, but we have seen this change in set point viral load increase by about 0.2 log in the last decade. And because this is a project done with CDC, they would like me to tell you that public health action targeting rapidly growing clusters may act as a counterpoint to this natural selection. So with that, I'd like to thank you for coming and thank my colleagues, especially Adiba Hassan and Britt Scathen, who did a lot of the early work, and then CDC colleagues Alexa Oster and Waleed Hanani, and then finally funding for this project from the NIH, CDC, UC San Diego CFAR, and also I have a grant from Gilead Sciences, but that's on hepatitis C, unrelated to this work. Thank you.
While I don't expect it to explain the increase in viral load, did you adjust for transmitted drug resistance, which would bias the results towards a lower viral load?
Every single person here was ART naive, and had pure wild type virus. So no, any compensatory mutation of the 108 codons, all stripped out. That being said, you see the same pattern in people with K103N and L90M. If you do the same analysis just in the drug resistance population, you see people with clustered, they have higher viral load than people who are unclustered. But we separated it out entirely by drug resistance.
So Joel, what was the end date of your analysis?
This was 2016. It takes a long time to get a paper.
Okay, so when were
CDC.
it's amazing that you've seen what you've seen with the increment. The drug pressure counts, right? I know this is natural selection, but
Yeah.
drug pressure is going to count. So the analysis when the integrase inhibitors kick in.
Hmm. So I didn't include here. So we looked to see if we could find an effect of test and treat. And I would say, do we see that slope in increase in viral load change in, say, 2010, 2011, 2012? And we didn't detect that effect. And we didn't detect any significant interaction between time and clustering on viral load. Now, is that just due to lack of signal, or is that the underlying driver of this natural selection isn't drug resistance pressure and it could be something else? We don't know. We were unable to get at a mechanism here, but we didn't find we didn't find signal that test and treat is driving this. That was our initial hypothesis, and one of our reviewers encouraged us to really get compartmentalized on it, and there's just not that signal. So we don't know why it's happening, but we see the evidence for it happening. All righty. Thank you all so much for coming.